Hello, everyone. My name is Jeffrey Gu, and today I'll be presenting my summer research that I conducted in Christopher Del Negro's lab in the Department of Applied Science. My summer research project examines the effect that opioids have on breathing rhythm. Before diving too deeply into my research, let's first unpack what opioids are and how they affect the body. Opioids are a class of pain relieving drugs that has been used recreationally for much of history. Recreational opioid use skyrocketed since the start of the opioid crisis when it began in the 1990s, and since then, opioids are responsible for a large percentage of the drug overdose related deaths, especially in recent years. During an opioid overdose, opioids cause a decrease in breathing rate, and death is often caused when breathing rhythm stops completely. So, to test the effects that opioids have on breathing rhythm, I examined a region of the brain called the pre-Botzinger complex, which was fairly recently discovered to be the breathing rhythm generator of the brain. Mu opioid receptors, which are activated by opioid drugs, are present in the pre-Botzinger complex. Thus, this project looked at how different concentrations of DAMGO, a synthetic mu opioid agonist, would affect breathing rhythm generated by the pre-Botzinger. Slice preparations of the pre-Botzinger can be recorded to track in vivo breathing in real time. The spikes in this slide represent a nerve output, which would be the equivalent of a breath in a living animal. To do this, this image is one of my experiments, and everything that's important is labeled here. So the glass section electrode is attached to the nerve. Specifically, this is the hypoglossal nerve, allowing for a successful nerve recording. And nerve recordings are what we use to measure breathing rhythm in vivo. This image also shows roughly where the pre-Botzinger complex would be located. From these results, we found Damco significantly reduced breathing frequency. At a concentration of 10 micromolar, on average, Damgo was found to reduce breathing frequency by 40%, signifying a decrease in breathing rate. This can also be seen with the graph on the left, which shows a reduction in burst frequency after Damgo is added. It also is important to note that burst frequency returns back to normal once the drug is washed out. On the other hand, at a concentration of 30 micromolar, Damgo was found to completely abolish breathing generation. This can be seen in the figure on the left, showing that burst frequency disappears when Damgo is added to the preparation. The disappearance of bursts signify that Damgo reduced breathing rhythm to a rate of pretty much nothing, which is likely what also happens during an opioid overdose. Through dozens of experiments, it was repeatedly found that Damgo significantly reduced burst frequency at smaller concentrations and abolished burst activity at higher concentrations. So, what this tells us is essentially that opioids' slow breathing rhythm and at very high concentrations leads to the stoppage of breathing rhythm itself. So, why is this research significant? Why do we care about it at all? While these results are not necessarily surprising, given that opioid overdoses cause breathing rhythm to stop, it provides us with a solid foundation for future experiments. Using this model, we can work to identify the specific cells and signaling pathways that opioids act on by selectively knocking out certain genes. Discovering the specific pathway of opioid induced respiratory depression may be the key in developing medication that can resuscitate breathing in a victim experiencing opioid overdose. While the opioid crisis may be a sociopolitical mess, this research has the potential in reducing the suffering of marginalized communities most affected by this crisis. So, with that being said, these are my citations for this slideshow, and feel free to reach out to me if you have any questions. Thank you for listening.